In this video, I'm going to explain very briefly what microRNAs are and how they work. MicroRNAs have only been discovered fairly recently, and they're a very important part in controlling not the way genes are expressed, but controlling the way messenger RNA is read off into proteins. So it's a post-translational level of control. So let's start off then with thinking about genes in a very simplistic way, that we generally have a promoter region. Again, this is very simplified. And then we'll call the open reading frame, which consists of a series of exons separated by introns. So usually what happens is that the promoter gets the gene to be transcribed into messenger RNA, and that messenger RNA is then processed to remove the introns, so you end up with the mature RNA, which drives protein synthesis. But now, some RNA is not actually involved in directing protein synthesis, including what we call microRNAs, sometimes just abbreviations to mir. These are about 22 nucleotides long, and they can be encoded either somewhere in the genome or quite often in one of the introns of a gene. How do they work? Well, they're transcribed into a hairpin structure like this, made of RNA. This then gets processed to get rid of the hairpin structure. So you've now got double-stranded RNA. Now, double-stranded RNA was discovered around about 1995 to do two things. One is it finds its way, one of the strands will bind to what we call the UTR, the untranslated upstream untranslated region of the protein gene, and it will inhibit basically what it does inhibits the production of the mRNA. It can also bind to the messenger RNA and label it for degradation. So in short, what it does is it reduces the production of the messenger RNA and therefore reduces the protein. Now, why it does that? All sorts of reasons, lots of speculation, but it does. So it's almost as if the protein gene, or somewhere else in the genome nearby, we've got a RNA being encoded which actually countermands or counterproduces the actual production of the protein. So it's like a way of fine-tuning the production of protein in the genome. So take-home message then is microRNAs are about 22 nucleotides long, and their role is to counterbalance the production of the protein for which they encode.